research begins with a problem. In the United States alone, over 20,000 biochemical substances have been found in marine life over the past 30 years. This causes extensive harm to aquatic wildlife, especially for a particular flatworm called planaria, a very common worm found in oceanic environments. When people dispose of medications unethically, they end up often in the ocean and end up harming not only the wildlife, but also the entire ecosystem as a whole. And moving away from the ocean, certain medications also pose extensive harm to human beings as well. The CDC states that over 106,000 deaths have been caused by prescription drugs in the past few years, and this number is projected to increase as well. So, throughout my work in the pharmacy, I took particular interest in a medication called pseudoephedrine, or rather referred to by its brand name Sudafed. And this medication, I quickly noticed, is a type of over-the-counter medication that is less accessible to the general public. It's a type of medication that we keep behind the counter, that people have to show their ID for, and has a variety of different restrictions that enables people to not access them as easily as other medications. This made me question, what permits these restrictions for Sudafed? And is it potentially harmful or addictive to the human body, potentially marine life as well? So hi, my name is Ella Mayer, and today I'm here to talk to you about cold medicine, worms, and wound healing. By first asking the question, to what extent does pseudoephedrine hydrogen chloride, or Sudafed, affect the regeneration of Digesia tigrina planaria? And basically, pseudoephedrine is a type of cold medicine, like I discussed. It's a very common over-the-counter medication. And Digesia tigrina planaria is a very commonly found regenerative worm that is very well known for its regenerative capabilities, whether in the ocean and also outside. Moving on to my goal of my research study today, breaking down into three major substeps. The first one being to first amputate the planaria. So in my study, I'm investigating the regeneration specifically as an endpoint in order to see the effect of PSC on its regeneration. So once I amputated the planaria, I exposed it to different concentrations of pseudoephedrine hydrogen chloride, and then I watched as the planaria grew back its head and as well as its tail under a microscope observation. Okay, so let's talk a little bit about the nature of brown planaria itself and why I chose to specifically study this organism in particular. Starting off with its composition. So planaria is a very fascinating creature because it is composed of 20% of something we call neoblasts, which are basically undifferentiated stem cells. It sounds like a complicated term, but essentially just the same way that humans basically specialize into different occupations, like how one can become a doctor or engineer, these type of cells have the ability to differentiate, basically specialize into specific types of cells in the body. So upon wound healing, like I see here, if you were to cut off the head of a planaria even into 272 different parts, every single one of those 272 parts would regenerate into a fully fledged worm. It would be able to regenerate its brain, head, eyes, oracles, and all, which is very, very fascinating. So one of my study that I'm particularly looking at is the formation of a blastema. So I talked a little bit about stem cells in neoblasts, but when the planaria is cut on the head mark, when it starts to regenerate back its head, you'll see the formation of something called a blastema, which is essentially just an aggregate of cells that form at the area of a wound injury in order to start to repair that area and rejuvenate the rest of its body. Okay, moving on to some pertinent surrounding literature regarding my topic. And today I'm going to be talking about establishing the gap in my research. Okay, so the first paper that I talked about is the effects of the dangers of ephedrine as well as pseudoephedrine as a nasal decongestant. Like I mentioned, pseudoephedrine is a chemical compound very commonly found in the pharmacy that works as a very effective vasoconstrictor. As this paper mentions, a vasoconstrictor is basically a medication that opens the airway to allow you for better breathing by constricting your blood vessels, and that is very effective for pseudoephedrine to do. However, on the alternative hand, it poses very severe adverse cardiovascular as well as neurological effects for both humans and potentially also aquatic organisms as well. So from this paper, I deduced two main things. The first one, verifying the dangers of PSC as a potentially harmful medication. And two, devising my hypothesis in general. So my hypothesis for this experiment was that increasing concentrations of pseudoephedrine would result in greater mortality rates and therefore a decreased regeneration rate for the planaria. Moving on to my second paper, I studied the effects of ivermectin exposure on the regeneration of a different type of species of planaria. This was a really quite interesting study because it was similar to mine, however, instead of using PSC, it used a different substance called ivermectin. And what ivermectin is a specific type of medication that is known for killing parasites. So throughout the study, they found that exposure of ivermectin increases mortality rates in the specific species of planaria. Or, very simply put, it kills the planaria. Okay, so from this paper, I deduced three main things. The first one, the use of planaria in my study. Planaria is very commonly used for toxicology studies, as well as pharmacological studies as well. And as seen in this previous paper, it was very effective for use in regeneration studies, as mentioned. The second thing, the use of the methodology. Since this experiment was pretty similar to mine, except for the difference in the type of medication, I was able to use a lot of the specific types of methods that they implemented in the experiment. And lastly, studying the different regeneration phases of the planaria. So from the formation of the blastema to the formation of the eyeballs and the brain, the development of its entire intestinal system, I was able to learn a lot about how planaria regenerates and how it recuperates from wound and stress. Okay, so this allowed me to establish my gap in the literature. Planaria has never been studied with pseudoephedrine before, and throughout its history, pseudoephedrine has been shown to be potentially dangerous to humans. But especially for a lot of medications, they end up in the ocean, harming a lot of aquatic wildlife underneath. And since planaria is a common aquatic organism that's found in fresh water as well as saltwater areas, pseudoephedrine can potentially cause harm for this organism as well. Okay, moving on to my methodology. Starting on with the type of experiment I decided to conduct. So for this study, I decided to conduct a mixed qualitative and quantitative correlational experiment. Sounds like a big word, but basically, I used both qualitative and quantitative factors in order to analyze the effect of the regeneration of the planaria. So qualitative factors, what did I take into account? I took into account the morphology of the planaria, the anatomical characteristics, and the development of different parts, and also the color of the planaria as it starts to grow back its parts. For quantitative variables, I took into account mortality rates, the regeneration rates, as well as the overall lifespan within the duration of my experiment. So for my entire experiment, I did it around a 20-day span, but for the first 5 days was mainly an observational period. So for that amount of time, I was just able to pick and choose the planaria and ensure that all of them are ready for the decapitation phase and ready for the treatment phase as well. And then on day 5 was when I amputated each of the planaria. And in this study, I used exactly 150 planaria, and when I cut them in half, it resulted in a total of 300 experimental subjects. Okay, moving on to the materials, sterilization, and also observing the planaria. This was really fun to do. I used a bunch of different materials. I didn't list all of them here, but these are some of the main stuff that I did use. So starting off with the digital microscope, this one was very, very helpful when I was able to look over the planaria in their petri dishes. So for the compound microscope, I used an OMAX 475 microscope, which zoomed in like a lot, so that was really helpful. But I would have to basically remove the planaria from its petri dish environment in order to actually see the different cells underneath. So that's where the digital microscope pretty much came in. It was just a lot easier to use since I can just put it right over the petri dish and watch its regeneration. Okay, next was the micropipette. This was really helpful. I learned this in AP Bio, which I thought was really interesting. But it was very good in using precise measurements. So for each of my petri dishes that I used exactly 7 milliliters of solution. So I wanted to make sure that it was as accurate and precise as possible. And lastly, of course, the planaria itself. So if you're ever asking where to find planaria, I found it at Carolina Biological Supply, which is actually available to most other people, but I helped find this website with the help of my mentor. Okay, moving on to the second step. I separated each of my petri dishes into 5 main groups, 3 experimental and 2 control groups. For the experimental groups, I started from a low concentration, then a medium, and then a high concentration of pseudoephedrine. And for my control groups, I used a negative and a positive control as well. So for my low concentration, it was 30 milligrams, then it went to 120 milligrams, and then 240 milligrams of pseudoephedrine. And then my negative control was just spring water with no addition of any other substance or medication. And then for my positive control, I used caffeine, since caffeine was proven throughout multiple studies in my literature review to increase rates of regeneration in the planaria. Okay, so here's my actual setup of my experiment. So you can see here, I did multiple trials because obviously the actual petri dishes are pretty small, and I did multiple trials so I can be able to incorporate and validate my results throughout the experiment. So this is the low, medium, and high concentrations of pseudoephedrine, as well as the negative control, which was just straight up spring water, and the caffeine positive control right there. And then of course everything else right here, and then these are the planaria. They're really, really small. I'll show them in the other picture as well. But for the decapitation period, I did them at day 5. So after the observational period, I decapitated the planaria using a 2-step process. First, I immobilized the planaria, putting it on ice and just slowing down its movement so it's a lot easier to cut it at a precise angle. And then second, I sterilized a razor blade and scalpel, and then I used a horizontal bisection to decapitate the planaria's head from its rest of its tail. So here's a little bit of comparison of the actual blade in comparison to the actual planaria. You can see here, the planaria is really, really small. So a lot of them fit in the petri dish, which was really great. But it was a little difficult in this process to actually cut off the head because I had to make sure that it wasn't moving because I wanted to cut it as precise as possible. All right, time for my results. I'll be talking about phenotypic differences, morphology, as well as differences and changes in the anatomical features of the planaria. Beginning with the control group. So this is under the OMAX microscope, so you can see the cells of the planaria very vividly, especially compared to the digital microscope. But for the control group, you can see the development of the planaria's tail is very distinct, and there's no alteration in any of its regeneration stages. This is the anterior side of the planaria, which is just its head, and you can see its auricles, which are basically its ears. The eye spots are also very widely visible before the experiment, so it looks pretty happy, which is great. And then the posterior, of course, is its tail. Okay, so this is when it gets a little gory. So after 1 day of exposure to pseudoephedrine, I studied exactly how the planaria would behave in response to this medication. So with the lowest concentration, which is the 30 milligram of pseudoephedrine, you can start to see the planaria start to coil around each of the solutions. This is present in each of the different concentrations of pseudoephedrine, but it's very easily visible in the 30 milligrams since the 240 is a little bit cloudy and you can already see that it's a higher concentration of pseudoephedrine, so it's a little more difficult to see. But the reason why planaria actually curls up is to protect its internal organs. Planaria, since it absorbs materials through a process called epithelial diffusion, any material that's around its surrounding environment will easily absorb into its body. And therefore, if it's a very dangerous substance, it'll start to curl up and start to protect all its organs in fear that something will happen to it or it will cause death. And over the 10 days. So for each of the different solutions, you can see drastic differences in the actual anatomical characteristics of the planaria. The eye spots are no longer visible. You can no longer also see the digestive system, as well as the auricles and its brain, the rest of its body. It's nothing is really left. It's just mostly just a mass and clump of cells. So what I particularly looked at is with the 30 milligram and compared to the 240 milligram. The 30 milligram, although it caused greater mortality rates in the planaria and a lot quicker of a death, you can see that a little bit of the structure is still intact in the planaria compared to the 240 where it literally just looks like a bunch of cells. Okay, moving on to the control variables. So this is how the regeneration of planaria should look like, obviously not in response to pseudoephedrine. So we can see the formation of the blastema, which again is a mass of cells formed during the early stages of healing. You can also see in this one the positive control, since it regenerated pretty quickly since it had the exposure of caffeine. You can see the formation of eye spots start to develop on the planaria. You can start to see that lobe shape of its head right there, in comparison to the negative control, which took a little longer to heal, but it still healed pretty well. And that white part is all the blastema right there. Okay, time for the statistical analysis. So I did two different steps for this one. I first conducted a chi-squared analysis, and then I also conducted a observation of the mortality rates throughout the 10-day span. So starting off with the chi-squared analysis, I used this formula right here and calculated the amount of active and deceased planaria at the day 3 mark. So I found that the chi-squared statistic is 181, meaning that there is a statistically significant correlation between pseudoephedrine and mortality rates, specifically at day 3 in the experiment. And the reason why I chose day 3 was because at that time, most of the planaria was already deceased in the lowest concentration of PSC. So from this chi-squared analysis, I deduced 3 main things. The first one, evidence of statistically significant result, meaning that there might be a possible correlation between pseudoephedrine and the regeneration of planaria. 2, I was able to reject the null hypothesis. And the null hypothesis for this experiment was that there was no correlation that exists. But through this experiment, I was able to find that there was certainly a correlation. And of course, I was able to establish an overall relationship between pseudoephedrine and planarian death beyond mere fluctuations in the sample. Okay, moving on to mortality rates. So I took the mortality incidence at day 3 of the experiment, as well as the average planaria mortality rate per day. So at day 3, you can already see most of the planaria is dead in the lower concentration of pseudoephedrine. And it's a similar thing with the average planaria mortality rate per day. So over the 10 days, when I took the average as well as the standard deviation of each of them, majority of the planaria died a lot quicker in the 30 milligram solution. Okay, and then I also did a little statistical calculation on how likely the planaria were to die within the first 3 days of the experiment. How quickly can it absorb into the planaria and start to cause its detrimental effects? So for the 30 milligram solution, I found that it had the greatest chance of likelihood at a 98% likelihood that the planaria will die within the first 3 days of exposure. Moving on to the discussion. So what exactly do these findings mean? Starting off with, the planaria will absorb very quickly via what we talked about earlier, epithelial diffusion. As you can see, the nature of the planaria starts to curl up and coil up, and this is a very adverse response to the very dangerous medication. So it did not react positively towards exposure to PSC, which is something that is very concerning, especially if these medications are wasted in the ocean years after years due to unethical disposal of these medications. Second thing, the lack of alpha adrenergic receptors caused excessive vasoconstriction-like effects in the planaria. It sounds a little confusing, but basically, alpha adrenergic receptors are a type of receptors that PSC targets in us humans. But planaria, as a very simple aquatic organism, lack these type of receptors. Therefore, it causes these excessive effects in the planaria. Okay, moving on to limitations and future directions. So the first limitation that I encountered was temperature fluctuations. Throughout the entirety of the experiment, I did take a thermometer in order to ensure that the temperature of each of the solutions were as consistent as possible. But of course, this could not completely rule out any small fluctuations in the temperature level. So for future research, I suggest that they do have a better method of keeping it in a housing container to maintain a more controlled and sterile environment. Also, light exposure was a very significant factor in my project. So in order to actually see the planaria under the microscope, I would need light. And the thing with planaria is that it does not like light. It likes staying in the dark. So in order to see it, it was a little difficult to kind of move it around and ensure that it wasn't moving away from the light source. And then also a single species focus. In this study, I used specifically Digesia tigrina planaria. It's a brown planaria, but there's also other species like white planaria. There's also black planaria as well. So this results in my study can only be attested towards this specific species of planaria. So for future directions, they could probably use different types of species to validate those results. Also, a lack of a detailed molecular analysis. I only had access to a compound microscope and a digital microscope as well. So with access to an electron microscope, for example, or better resources and in a lab setting, most likely, it would be a lot easier to see the different cells and potentially even the receptors or even the genes of each of the planaria and how it changes as a result. So sample size constraints was another thing as well. I used a total of 300 experimental subjects, but for the future, to validate these results, more experimental subjects in planaria will need to be used. Okay, time for implications. The first one I talked about was ecotoxicological relevance. This basically means that planaria living in its normal atmosphere, which is in the ocean and whether saltwater or freshwater, it can very easily be exposed to any of these dangerous medications. So relevance in this sense is very important. Next one, fundamental biological mechanisms in regenerating tissues. Studying the exact mechanisms that planaria employ in order to regenerate and recuperate its cells is very important, and it's still a sort of topic that needs to be studied. So this experiment validated a lot of those different types of mechanisms. And the last one, public awareness of pseudoephedrine drug overdose. Drug overdose causes deaths across the United States and even the world. So it's something that is very prevalent in society today and something that we definitely need to cause more attention to. And so why should you care about this? You should care because over 20,000 biochemical substances have been found in marine life. You should care because 106,000 deaths have been caused by prescription drugs alone in the United States. You should care because you must dispose of your pills in an ethical manner in order to prevent both dangers towards humans and also dangers towards aquatic marine life creatures as well. Thank you.
Okay, for your first oral defense question, how did your review of the methods used by scholars in the field inform your selection of a research process that is aligned with your project goal?
Okay, this is a really good question. I did talk about one of my specific literature reviews that I reviewed. That's the second paper that I looked at. It was the effect of ivermectin on the regeneration of planaria. And that study really helped me deduce a specific type of methodology. It wasn't even just the materials I used in that section, it was being able to cut the planaria. And the best way to cut it and the best way to observe its regeneration stages, those are the specific types of steps that I utilized from that study in order to apply to my own and understand or gain a better understanding of its regenerative process.
Okay. For your second question, how do your findings provide directions for future research in the field?
Okay, so but this kind of goes on with future directions as well. But my research essentially found, or very in simple terms, that increasing concentrations of PSC causes harm, but particularly within the lower concentration of pseudoephedrine. And pseudoephedrine, I don't think I mentioned this earlier, but it's a medication that is a precursor for methamphetamine. So it has a history of being dangerous towards humans, but I really wanted to discover or study how it would affect wildlife as well, in terms of aquatic wildlife, since it was a very prevalent issue that has been disposed of in aquatic environments and oceans and that as well. So future directions, I would think, is probably to increase the sample size, try different organisms that could also potentially grow in the same type of freshwater environment that planaria do, and also to use a better molecular analysis in order to deduce these specific types of receptors, the cells, and the development of every single step and phase of the planarian regeneration.
Okay, and for your final question, how did your expert advisor facilitate your deeper understanding of the research process?
Oh, okay. So I worked with Dr. Anna Cunningham from USC, and also she's the pharmacist or pharmacy manager I work with at CBS. And she helped me a lot with my process in terms of actually understanding what pseudoephedrine is and the chemical compounds. So I could obviously search that online, but it was just a lot easier to be able to understand from a professional in that field who can very easily explain to me the chemical compound and exactly what it is. So pseudoephedrine, what she explained to me, pseudo means basically an alternative of that version. And ephedrine, it originates from a specific plant called ephedra. And ephedra is a plant that has a lot of recognition for stimulation, neurological stimulation. And that is one of the dangers of why pseudoephedrine can obviously be a very effective vasoconstrictor, but she also told me that it can be very dangerous towards humans as well, especially in high concentrations.
Okay, thank you.
Thank you.